Here, let's talk about reactions of monosaccharides. Monosaccharides are the simplest carbohydrates found in nature, and they have a general formula CaNH2ON. To understand what kind of chemical reactions that a monosaccharide undergo, we must first figure out what is the functional group that the monosaccharide possesses. Well, most of the monosaccharides have an aldehyde or ketone as a functional group, hence the chemical reactions that the monosaccharides undergo will depend on these functional groups. These reactions are oxidation to give acids, reduction to give alcohol, reaction with phenyl hydrazine, formation of ester with phosphate group, and dehydration, that is removal of the hydroxyl group. Let's see each of them. Oxidation. The aldehyde group or the keto group or the terminal hydroxyl group can be oxidized to form acids. In case of glucose, the oxidation of aldehyde group leads to formation of gluconic acid, while the oxidation of terminal alcohol group leads to formation of glucuronic acid. Reduction. Reduction of aldehyde or keto group results in formation of alcohol. Reduction of glucose gives sorbitol, galactose gives delketol, mannose gives mannitol, and fructose gives mannitol and sorbitol. Reactions with phenyl hydrazine. Under acidic condition, monosaccharides can react with phenyl hydrazine to give osazone. The osazones formed by different monosaccharides have different crystal structure. For example, glucose and fructose gives needle-shaped crystals, lactose and galactose gives fluffy ball-shaped crystals, and maltose gives sunflower-shaped crystals. Esterification. Glucose can be esterified to give glucose 1-phosphate or glucose 6-phosphate. Dehydration. Monosaccharides, when treated with concentrated sulfuric acid, undergoes dehydration by releasing 3 molecules of water. The product formed during dehydration are called furfurols. The furfurols so formed are chemically active and can react with phenolic compounds like alpha-naphthol to give colored products. Tautomerization. This is a process in which there is a shifting of hydrogens from one carbon to the other to form enediols. The enediol formation is usually facilitated in the alkaline environment. For example, glucose can be converted to fructose by its intermediate enediol in an alkaline environment. Let's talk about disaccharides. The most common oligosaccharides found in nature are the disaccharides. The disaccharides consist of 2 monosaccharide units linked together by glycosidic bond. The disaccharides are crystalline, water-soluble, and sweet in taste. If the disaccharide has a free aldehyde or keto group, then it's known as the reducing disaccharide. Example, maltose and lactose. If the disaccharide does not have free aldehyde or keto group, then it is known as non-reducing disaccharide. Example, sucrose. Now take a pause and look carefully. As far as the structure of disaccharide is concerned, they are represented in pyranose or furanose form. If you have a structure of glucose, glucose would look something like this, and this is the alpha glucose, while this is the beta glucose. It is the first carbon atom that has the aldehyde group, so the first carbon of glucose has the reducing properties. If this bond is free in disaccharide, then the disaccharide will be reducing, otherwise it will be non-reducing. In a similar way, imagine a structure of fructose. It's the second carbon which has the ketone group, so the second carbon must be free in order to have the reducing properties. So let's talk about different disaccharides. Maltose. Maltose is formed by two molecules of glucose linked together by alpha 1,4 glycosidic bond. The second molecule of glucose still has a free aldehyde group, which gives maltose its reducing properties. Maltose can be hydrolyzed into two molecules of glucose by the enzyme maltase or by dilute acids. Cellobiose. Cellobiose is similar to maltose, but instead of alpha 1,4 glycosidic bond, it has beta 1,4 glycosidic bond. Cellobiose is formed by hydrolysis of cellulose. Sucrose is commonly known as sugar. It is made up of glucose and fructose joined together by alpha 1,2 glycosidic bond. It should be noted that in sucrose the glucose has alpha structure while the fructose has a beta structure. Lactose. Lactose is commonly known as the milk sugar. Lactose is formed by galactose and glucose linked together by beta 1,4 glycosidic bond. In lactose, the glucose still has a free aldehyde group, hence lactose is a reducing sugar. Hey everyone, quick biochemistry basics here. Let's talk about Fehling's test. Fehling's test is used to distinguish monosaccharides having aldehyde or a keto group. Monosaccharides having aldehyde group gives positive Fehling's test, while the monosaccharides having ketone group gives negative Fehling's test. However, there is an exception over here. Monosaccharides having alpha hydroxy ketone like fructose can also give a positive Fehling's test. Fehling's reagent consists of two solutions which are known as Pheling A and Pheling B. Pheling A is an aqueous solution of copper sulfate which is blue in color, while Pheling B is an aqueous solution of potassium sodium tartrate along with strong alkali such as sodium hydroxide. The final working solution is prepared by mixing equal volume of Pheling A and Pheling B. When monosaccharides are mixed with Pheling solution and heated at 60 degrees Celsius in a water bath, the monosaccharide is oxidized to acid while the Cu+2 ions are reduced to copper oxide. The copper oxides form is reddish or brown in color and can be quantified easily to find the amount of aldose monosaccharide present. This is a very common test used to detect presence of glucose in urine as a positive indication of diabetes. Let's talk about Tollen's test. Tollen's test is a test used to distinguish between aldehydes and ketones, and in biochemistry, it's a test used to distinguish between monosaccharides having an aldehyde group versus monosaccharide having a keto group. Only the monosaccharide having an aldehyde group reacts to give a positive Tollen's test. However, there is an exception over here. This test cannot distinguish between aldehydes and alpha hydroxy ketones like fructose. Tollen's reagent. Tollen's reagent is made by mixing silver nitrate with sodium hydroxide, and this reaction results in formation of red-brown precipitates of silver oxide. The red-brown precipitates of silver oxide are then reacted with ammonia, which results in formation of soluble diamine silver complex. The soluble diamine silver complex is also known as ammonical silver. When an aldehyde or an aldose monosaccharide reacts with diamine silver, the aldehyde group is oxidized to acid while the silver ions are reduced to metallic silver. The reduction of silver ions to metallic silver results in a formation of silver mirror on the surface of the test tube, which indicates a positive Tollen's test. Because of this reason, Tollen's test is also known as silver mirror test. Let's talk about Benedict's test. Just like Fehling's test and Tollen's test, Benedict's test is used to differentiate between monosaccharides having aldehyde group versus monosaccharides having a keto group, with an exception like alpha hydroxyketones like fructose, which will give a positive Benedict's test. Benedict's reagent. Benedict's reagent consists of copper sulfate, sodium carbonate, and sodium citrate. The sodium carbonate provides an alkali environment. Now, in alkali environment, aldehydes or aldose monosaccharides like glucose are converted to powerful reducing agents like enediols. Enediols can reduce copper ions to copper oxide, which can either be green, yellow, orange, or red in color. The color of the precipitate so obtained gives the estimate about the amount of reducing sugar present. This has an important role in estimation of glucose in urine as a positive indication of diabetes. Let's see the role of sodium citrate. The copper sulfate and sodium carbonate can react with each other to form a precipitate of copper carbonate. Sodium citrate prevents this reaction by reacting with copper and forming copper citrate complex. The copper citrate complex is very stable and have a long shelf life. Hence, the reagent can be prepared in lab and can be kept for a very long time.